The pharyngeal clefts or arches are in the pharyngeal part of the digestive tract. They're behind the oral cavity and anterior to the esophagus. So this is in the throat area. And they look like pairs of pouches that push outward from the lateral walls of the pharynx. So they're continuous slit-like passages connecting the pharynx to the exterior. And as far as what happens to these clefts and slits, it varies a lot from each taxonomic subgroup. But you can see here the pharyngeal pouches and the dorsal nerve cord in yellow, with the notochord in front of it in pink, and then the postanal tail that extends beyond the anus. Those are all chordate fishes, or features, not fishes, features. In your fish-like vertebrates and in the juvenile amphibians or tadpoles, the walls of the pharyngeal clefts will become gills, and that's how they exchange gases with the water in their blood. In the adult amphibians and the amniote tetrapods like reptiles, birds, and mammals, the anteriormost cleft becomes the auditory tube and the middle ear chamber, and the other clefts disappear after they become parts of glands and lymphatic tissues in the throat region. Now the subphylum Tunicata, or the sea squirts, they're underwater sac-like filter feeders, and they have an in-current siphon and an ex-current siphon. Most of them feed just by filtering the seawater through their pharyngeal slits. They do have a notochord during early development, but they don't have myometric segmentation throughout their body and tail as adults. They begin life as a mobile larval stage that looks like a tadpole, and later they become barrel-like and sedentary as adults. Their primary source of food is plankton, and that plankton gets entangled in the mucus that they secrete from the endostyle. Their pharynx is covered with miniature hairs, and this allows the consumed plankton to go on down through to the esophagus. Their guts are U-shaped, and their anuses empty directly to the outside environment. They're the only animals able to create cellulose. But their blood is what's interesting. It has high concentrations of the metal vanadium and vanadium-associated proteins, and they can concentrate the vanadium up to a level of 1 million times that of the surrounding seawater. And these specialized cells concentrate heavy metals, which are then deposited in their tunic. And this is what they look like. You can see the in-current siphon and the ex-current siphon there.